Hello everyone and welcome to our video series Genomics for All, GFA, from MyBiome Therapeutics. So for those of you who missed the previous editions, previous episodes of this, we spoke about healthcare, agriculture, renewable energy, synthetic biology, animal biotechnology and so on. So you can view our episodes in our YouTube channel Genomics for All. Today what I'll do is, we have been talking about healthcare a lot, and diagnostics, as you know, is a very important part of preventive as well as curative healthcare. So in this, today, genomics is being integrated in a very holistic way in the actual diagnostics field itself. So for example, if you take a lab which has imaging and pathology, and there is a clinical history, electronic medical records, electronic health records, both preventive and curative, now you can integrate genomics on top of this, and this is being done. What I'm saying is being done in developed countries, and India is catching up in a big way. If you add genomics as a layer on top of all this, what you can do is more accurate prediction of disease, more accurate management of treatment, etc. So that is where integrated diagnostics, now we are beginning to call it integrated diagnostics, where genome-based diagnostics is being integrated into the routine diagnostics, as you call it. Where are the applications of all of this? We've been talking about rare diseases in a big way in this series because that's where genomics has benefited patients and science in general in a very significant way. So in rare diseases, for example, if you have the clinical history of a child and then you add genomics, you integrate genomics, the diagnostics of that particular disease, rare disease, is way much accurate than clinical history itself. Similarly for cancer, in our company we do a lot on cancer, on rare diseases. We work on kidney diseases, wherein it has been clearly shown that clinical history along with genomic data can add significant value to your whole accuracy of the diagnostics. And then we work at MyBiome, we work a lot on cholesteremia, hypercholesteremia and cardiomyopathies, etc., wherein this has been clearly shown that integrated diagnostics or genome-based diagnostics is the way going forward. So what we talked about in the previous episodes is also the applications of artificial intelligence, machine learning into genomics. Now imagine you add artificial intelligence, machine learning into genomics and then add this into the whole integrated diagnostics. So the power of everything becomes so much that now you are able to predict and prevent and treat a disease with much more confidence. So these are some of the examples that I thought I'll bring in. And another thing that is coming up in a big way in integrated diagnostics is multiomics. So in our series we talk about one aspect of multiomics, which is genomics, and then there is transcriptomics, there is proteomics, the global study of proteins in a cell, which is responsible for disease, as in healthcare and many other applications. So multiomics, for example, is the study of genomics, proteomics for protein, and metabolomics. And the combination of all of this, genomics, transcriptomics, proteomics and metabolomics, is what we call as multi-omics. Now, as the name suggests, it's a combination of many omics methods, as I said, genomics, proteomics and metabolomics. Now, how does all this connect to what we do at MyBiome? Interestingly, and today I thought I'll bring that here, interestingly, we started with next generation sequencing as a platform for genomics. And technologies have evolved now that next generation sequencing technologies that we use routinely for genomics are now being leveraged for studying proteomics and metabolomics to that extent. So what companies like Illumina, Thermo have done, Oxford Nanopore have done is very interesting, that next generation sequencing, that core remains the same, and now there are many different interesting ways wherein you can tag DNA to a particular protein, and that's the barcode of the protein, and thereby sequence the DNA, and as a result you interpret which protein you are sequencing. Very interesting ways Oxford Nanopore has come up with proteins directly entering the nanopore and getting sequenced directly. So now in our company, as you all know, we have MySeq, NextSeq 2000 and NovaSeq 6000 and Oxford nanopore machines. So imagine the power that we have now in our hands, not just genomics, but using the same machines, we can add one more technology to study proteomics, another technology to study metabolites, wherein DNA barcodes are added to specific metabolite like glucose, or let's say it could be a sugar, it could be a hormone, etc., and studied directly using NGS. And NGS has the power to do it much more high throughput way. So the entire study of genes was possible using NGS. Now the entire study of proteins and metabolites is possible by using NGS again, and thanks to all these innovations that are happening across the globe in using multi-omics as a platform to detect, prevent and treat a disease, and that's where the future is. And we at MyBiome have already the platforms that are required for this, NGS platforms, and happy to connect with any of you. It could be a clinician, researcher, a pharma company, biotech company, diagnostic lab, or anyone from the government who is interested in this technologies, the integration of all these technologies to finally apply to what we call as integrated diagnostics. Thank you everyone for supporting our video series Genomics for All, and we'll come up with some very new and interesting topics to discuss in the coming episodes, and thank you all.